Please, thank you. Take your seat and welcome to this afternoon session. The coffee break will be running until the end of the day, so no rush right now. So welcome to this session. My name is Giuseppe Lauria, and actually I'm a neurologist, essentially a doctor, and very pleased to chair this session on precision medicine. I have to say that as a doctor, actually medicine tend to be precise since ever. What has been changing very quickly in the last few years is likely the targeting of the actions the doctors take, which are intended to be precise. And this has very much to do with many of the things we have heard today, which is related to a concept that I would like to share, that the fact that the advance in the field of medicine for the benefit of patients will come when we will move from the current classification, syndromic classification of the disease to a biology-driven classification of the disease. And I'm pretty sure that today the great speakers that we will have will enlighten us on that. So I'll leave the microphone to my friend and co-chair, Professor Amalia Gastaldelli, to introduce the first speaker, please.
Good afternoon, everyone. It's a great honor and pleasure for me to be here today. I think it's a very interesting and well-organized meeting. I'm Amalia Gastaldelli. I'm working at the CNR, where I'm leading a group for studying all the metabolic diseases. I started as an engineer, and I graduated here in Padua, so I'm glad to be here. I feel to be back at home. And without further delay, I think we can start. And it's my pleasure to welcome Professor Matej Oresic. And Matej is a professor of medicine with a specialization in system medicine at Örebro University. He's also a group leader in system medicine at University of Turku, and serves as a member of the board of directors of Metabolomics Society. He does a lot of studies in system medicine, and I'm glad to hear and looking forward to your presentation, Matej.
Thank you for having me here in this beautiful city and historic city, and a great event that I enjoyed this morning. So first I would like to thank to many members of my team. I actually have two groups, one in Finland, in Turku, one in Sweden, Örebro, and of course many collaborators and many funders that have supported our research over the years. So first, just a small introduction of the concepts. So what we study is mainly metabolites, so-called metabolomics, and the reason why we study that is because metabolites are very informative about the phenotype, basically meaning what in a clinical setting means certain properties of organism, like a certain disease status, etc. So metabolites, so all levels are important, but metabolites are highly informative in certain contexts, which is also highly relevant then to the personalized medicine if you try to do the phenotyping of disease, etc. So this is not only layers. One layer that is highly important, and this is hopefully I will show and convincingly show today that these exposures are very important if you try to consider personalized medicine. So exposures, of course, is everything that we get exposed throughout the life course. How to quantify, we don't know yet. So I'm sure that AI will give have a very important role there. But it means all kinds of exposure. So it's a social status, could be ecosystems, could be lifestyle, nutrition, drugs you're taking over the time, and of course also like a chemical exposures. It could be air quality, but it could be, for example, also like exposure to environmental chemicals. And this will be the main theme that I will be explaining today. So how the chemical exposures, which are of course a very relevant topic today with all the pollution, etc., how they impact certain diseases. And that is called the exposome. So when we think about the progress of a disease, complex disease, it never just appears. So there is a diagnosis that appears, but before that, a lot of things can happen over the course of the life. At the early stages, okay, no. So at the early stage, I can actually use it here, yeah. Okay, so early stages, you may have predisposition genes, and then environment will interact with the genes, so you have gene-environment interaction, so you will have certain patterns appearing over the course of life that could then lead to identification of prognostic markers that can help you predict the disease or at least assess the risk. And then at some point you will get to the threshold. So in order to be able to detect disease, so you have to have very sensitive methods to do that. And then if you know the mechanisms in these early stages, then of course, ideally and hopefully, you would have also the treatment that could help change the course towards disease. So we have an early prevention. And of course, how to study this is very difficult, because if you are talking about a lifetime, you're talking about 20, 30 years of exposures and interaction with the genes and the host. Yeah, you have to actually then have samples available from this life course, right? From birth, possibly, or even before birth. So you have to set up studies that could actually follow this disease status. And I will focus today on one specific case which has been going on in my research for actually almost 2 decades, and that's on type 1 diabetes. But one fact that is important and relevant also for the future discussion of this topic is there has been very clear trend of increase of immune-mediated inflammatory diseases since the 1950s, 1960s. So this does coincide with the industrialization, changes in the environment. So there is some correlation, but of course we don't know if this is really causative. But there is a fact, there is increase. And I will talk specifically now about, give you a story about type 1 diabetes. So this is caused by progressive loss of insulin secreting capacity due to that of the beta cells in the pancreas. So there is a genetic component to type 1 diabetes. It's called, so 80% of subjects who have type 1 diabetes, they have certain immune system genotype related risk, so it's HLA locus. However, out of those who have this risk, only 3 to 7% get diabetes actually. So there is clearly also a very substantial environmental component to the disease. And the appearance of islet autoantibodies, or so-called seroconversion, is one of the early signs of the disease. And here is just to show example, one girl who was in one Finnish cohort, developed diabetes at the age of 9, but already had antibodies at the age of 2, and actually got 2 antibodies, which actually it's almost certainly then meaning that she will get a disease eventually, but it took about 5 years before she got it. One has to keep in mind that there is no signs of any disease, even when you have antibodies, so there is completely unknown. So basically, for the girl and her parents, the disease just appeared at 9 years of age. There was nothing before, any signs that could suggest that she is on the way to the disease. So we have studied metabolites, small molecules, in a Finnish cohort that was about 10,000 children, and this is an ongoing study, so this is a very old study of ours, but it was the very first one that showed some of the metabolic changes in type 1 diabetes. So it's 10,000 children, so since 1993, about 20% of all children born in Finland were screened for this study for HLA risk. Those who were at moderate to high risk were then invited to be included in this study. So there was very low dropout, and so a certain number of children developed disease. Samples were collected every 3 to 6 months since birth. So this is a lot of samples, and now you can go, for example, say who developed diabetes later on, and then you go back to the early days of samples and see is there anything there. So that was the design, comparing those who developed disease and those who didn't. But basically what we found was that there was a very substantial decrease of many phospholipids already before autoimmunity, and also some triglycerides, that is the kind of dietary lipids. But very interestingly, these phospholipids were even found in the cord blood, meaning at birth, to be decreased in those children who later, 5 to 10 years later, developed type 1 diabetes. So the question then is, of course, so that must be that this risk phenotype, lipid phenotype, is already there in early life, even possibly in utero, because that was seen already at birth. So this may have some role also in pathogenesis, but the question is, what is the cause? So what could explain these sort of changes already in the early life? So there is one interesting data which is very, very relevant to the discussion here, and that is that actually in the last 20 years, the incidence of the type 1 diabetes in Sweden and in Finland has been actually decreasing. So it has been going up over the decades before that, and then suddenly there is a trend of decrease, and it's actually, I mean, there seems to be now in this figure from the JAMA paper that there is one blip, but now, so actually it is, the trend still continues going slightly down or stable. And the same has been seen also then in Sweden. There was actually, curiously, just one jump that happened during the first year of COVID, and that was a very elegant paper in JAMA, showed that this is nothing to do with COVID. It has to do with the school closures for two months during the first year of COVID. And then after, in the second year, when the schools remained open during the COVID, actually there was no jump. It went back to the normal. So just a curiosity. So what could be explanation for this factor? So obesity has not changed. So that was one of the factors that was considered, still debated whether this is real or not. Enterovirus infections have a role in initiation of autoimmunity, but actually infections over the years has actually increased. So they do not explain the drop in incidence. So diet has not changed substantially over the last decade either. So this is not able to explain the incidence. However, there are certain compounds, the chemicals, environmental chemicals, that are very common in household and industrial products that have been introduced in 1950s and has been in wide use. But EU has started to regulate this much stronger over some of the major ones like PFOS and then also more recently PFOA, so that just within the period of last 20 years, actually the levels of these major PFAS have been actually decreasing in the environment, particularly in EU. So these kind of compounds, basically what they are, so they are used for like fire coating in the dishes, for the cooking, cookware, and today you can buy products that is said PFAS free, for example. And they basically, but it is like fatty acids, but they are replacing the hydrogen with the fluor. So they behave a bit like lipids, they have been shown to have a lot of impacts on the immune system, on the metabolism, so there's a lot of research going on, and it's actually even more going on right now, so increasing interest. So what is basically happening with PFAS, they have a long half-life, so they remain in the body for 5 to 10 years, so you cannot get rid of them. So that is the problem. So many of the compounds, the chemicals from the environment, you will excrete through urine or stool, but not so with PFAS. They will stay in your body, and the reason is, one of the reasons is that actually they utilize the same transporters in the gut as the bile acids, which are very important molecules for the regulation of lipid metabolism and glucose metabolism, homeostasis, so they play an important role. So they basically interfere with systemic bile acid metabolism, and so they re-enter this so-called enterohepatic circulation and stay in the body for that reason. So a very small fraction of PFAS actually gets secreted. So we have studied this, and just very briefly, so there's a study of children. Again, it's a longitudinal study, a bit smaller, but there was also stool collected and then the blood, and we studied the bile acids and then microbiome. And basically what we found, and this is now not a PFAS study, it's a bile acid study, so just to show that actually bile acids have a role also in type 1 diabetes. So what we found, that we identified several microbes that are metabolizing, that are actually metabolizing bile acids, meaning that so-called secondary bile acids that are produced by microbes, and they seem to be decreased in children who develop 2 or more antibodies, meaning that they will progress to diabetes later on. And then we found that actually this correlates with the secondary bile acid levels, so these are the certain bile acids that are produced by the microbes. So clearly the before the autoimmunity develops, we already see the disturbances in the bile acid metabolism. So in short, these systemic bile acids are associated with gut microbes, and they are altered in progression to autoimmunity. So that can explain possibly also disturbances then in the lipid metabolism. So the next, next to last story, we studied also then how do PFAS impact in mothers, the exposure, maternal exposure during pregnancy. How does it impact the lipids in the newborn children? So this is a mother-child cohort, about 250 mother-child pairs, mother-infant pairs, and we studied the metabolites and PFAS exposure in the mothers, and then look at also the autoimmunity later on in the newborn babies, and also the lipid levels in newborn babies. So what was interesting, so we did sort of partial correlation network analysis. We simplified the representation of the data, so we didn't look at the individual lipids or metabolites, but the clusters, so representing groups of metabolites. But the main finding here was, so we have PFAS measured in the mother, so this is orange, then we have maternal clusters, purple, and then you have the babies, so the lipid clusters. So, but basically the main finding was that we had a lot of PFAS here that were negatively correlated with the lipids in the newborn babies. So maternal exposure correlates much more than actually than the PFAS exposure in the mothers with the maternal metabolites. And these are all negative correlation. And if you look actually in the lipids that have been found associating with the PFAS exposure, it turns out to be the same kind of lipids that we found also exposed in the type 1 diabetes. So that actually, at that time we didn't actually have the hypothesis about type 1 diabetes, but now we started looking at it a bit more in detail. And indeed, so there was a clear correlation to these just models, regression models to show that basically various lipids of interest, they clearly correlate with the quartiles of PFAS exposure for different kinds of PFAS. And most importantly, we also found that you can actually develop a model just using the maternal PFAS exposure to actually predict which children will progress to antibody positivity later in life after they are born. So basically, maternal PFAS exposure seem to have a predictive value in terms of who will develop diabetes and who not in the offspring. And what is also interesting here that the top two PFAS, that they are the ones that are on the top list now also for being banned in Europe. They are still not, but I think that there is now already also discussion about the universal ban of PFAS production, not just in Europe. So now for the very last study, just want to highlight something that goes even further into the origins of the disease. We just published this paper in Lancet Planetary Health. And that is actually looking at the in utero exposure to PFAS in the human fetus. So this is a collaboration with Paul Fowler in Aberdeen, who has set up a fairly unique bank of human fetuses from the weeks 12 to 19 of gestation. This is from elective abortions, so these were normal fetuses. So here we had the 78 levers, and the levers, of course, are important because there it's also, this is the bioaccumulation site for the PFAS also in the fetus. Placenta actually can pass very easily the PFAS into the fetus. So because it uses the same transporters for bile acids as they are in the gut. So they are the only two organs that have these transporters. So this is quite a actually interesting and relevant finding. And so we measured various metabolites, but we also measured PFAS in the human fetuses, different gestation ages. And just to highlight what was the main finding here, was that we found, for example, that some of the key bile acid metabolites, including so-called C4, which is important for the biosynthesis, initiation of biosynthesis of bile acids, they actually correlate very, very strongly in the liver with some of the major PFAS. So like PFOA is one of the old ones. And what is also interesting is that this C4 was considered not to even exist in the fetus, because this pathway is not meant to be there yet in the fetal liver yet. But you can see here that actually, yes, it seems to be that the exposures, chemical exposures, seem to drive this pathway and synthesis of this important metabolite, central metabolite in the synthesis of bile acids. So this was actually then also confirmed using the RNA sequencing. So many of the genes like FXR and also like CD36, which are important in lipid metabolism, they were highly upregulated in the human fetus. So more or less in line what we found with a measurement of various bile acids. So in conclusion, so we showed in this study that direct evidence that there are in utero effects of PFAS exposures on specific key hepatic products that regulate lipid metabolism, glucose homeostasis. So they have a very major impact on the metabolism in the developing fetus. So before the babies are born, they already have a clearly disturbed core metabolism. And that some of these metabolites correlate later in life with various diseases, metabolic diseases, like, for example, fatty liver disease. So it means that already before they are born, babies acquire certain phenotypes that seem to correlate with metabolic disease decades later. So that basically means that I think that these findings may also give us some ideas about the developmental origins of many other diseases. So with that, I would like to thank you for patience, and I would be very happy to answer some questions.
Thank you, Matej. While we are waiting for some question, I have one for you. You have beautifully shown that, and I believe these were non-excessive exposure to PFAS, right? These were general population, not highly exposed because of a contamination of the environment, correct? And my question is, considering that this is an AI session, you beautifully shown that you had to use AI to integrate the multiple omics that you use in your study, because you use metabolomics, lipidomics, transcriptomics, and probably some genetic as well. And what is the role in your study about AI? And this, of course, not only descriptive, but could be also used as causative. There are causative models that can be done on your data, or just...
Well, there is actually several. I was a bit implicit here because I always try to focus on this, on the biology, but of course there are several levels. One is, of course, clearly the predictive models. So because you try to bring in a lot of data, and then you will use various kinds of approaches to derive the models that should be explainable also, of course. So that's one constraint when we think about AI. So that's one thing which is fairly obvious that has been going on and applications for many years. The other is the networks, of course. So there is actually underlying methods here for the network generation. They are not simple correlations, because you have to account for the spurious associations as well, etc. So there is certain Gaussian models that are going into in this modeling, because important thing is to identify basically what are the true associations and what are not. The third, which is actually even more hidden here, it is but I didn't want to, because it would take me a little bit off track. In the liver, human liver, we use so-called genome scale metabolic models. So we actually model using stoichiometry of the metabolism, and for every microbial species. And then using this, you have to, of course, constrain the model in a proper way, and then derive the model and to be able to predict which metabolites are produced by which microbes. And that basically tells you exactly what is the link between the specific bile acids as a product of microbes, and then identify the microbes that are actually producing them. So that is then model-based. So that is, I think, something where AI is extensively used. And then the fourth one, which was not applied here, but we just published a paper last night that actually is using that. It's this so-called mediation analysis. That's where we try to find basically what is cause and what is effect. It's a hypothesis generating. So it's not going to tell you exactly that this is the cause and what is effect. And then the idea there is to identify which let's say, factors such as metabolites are actually mediating the effects of exposures and leading to specific phenotypes. So that's another, so I gave 4 examples. So, and if you dig deep enough into the methodology, you will identify some of the AI components as well.
Any question from the audience? Please.
I can hear you.
Mike, wait, hold on, it's arriving.
Do you hear me? Yes. Too much, probably. Have a question. What about the use of drugs in depression in pregnancy? Did you make any study on that? Because it's a huge problem in the last years. The use of medicines during pregnancy for diabetes, hypertension, depression.
Yeah, so we actually, yes, we are. I'm actually coordinating one EU project that is looking at these early life factors and the long-term disease outcomes in the children. And maternal depression is one of the topics. We are also looking at the microbiome and link to the depression, and how does then this impact the long-term outcomes, like including neurocognitive outcomes. In fact, we have in 2 weeks a paper in Cell coming out on the topic of the ADHD and autism in relation to the microbe.
Wonderful, thank you, because I'm the president of an association, volunteering association for the prevention and treatment of depression in pregnancy and postpartum. And we are looking for the opportunity to trace what is going on in the baby after in the newborns, to understand if the prevention, meaning many kind of treatments, not only drugs, not only psychology, psychotherapy, but also natural therapy and music therapy and other good practices can give the opportunity not to have this strong impact on the cognitive impairment of the babies later, and also of the mother, because when the mother is ill, the baby is ill, is sick. And
yes,
when
so
we
are working on this kind of data, and one of the, I think from the modeling perspective now, what is making it challenging is because you have data that is both like continuous, and then you have discrete data, and some of the reliability of some of this data, which is more descriptive, is not as high as some of the quantitative, and how to bring this together. This is, I think, exactly probably where AI would be very useful.
Okay. Wonderful. We are also thinking.
Thank you. I'm sorry.
Too much.
I think
later.
you can
Thank you very much.
Oh, thank you very much again, and I'm pleased to invite on the floor Professor Jens Richter. Professor Richter is Professor of Engineering Science at the University of Oxford, with a joint appointment with the Institute of Biomedical Engineering and the Nuffield Department of Medicine. He's a group leader at the Big Data Institute and affiliated to the Ludwig Institute of Cancer Research at the Wellcome Center as an adjunct member. The title of his talk is Beyond Genomics, AI as an Enabler for Next Generation Pathology. Thank you.
Thank you for your welcome, and thank you for the invitation here. I think it's a wonderful conference to really broad list of topics in AI. And I was also think there were some really nice poster presentations that sort of reflect also what's going on and where are new research opportunities. I wanted to talk to you about the topic of pathology, and the chairs basically started the session with that we should sort of think about subtyping disease and subtyping patients. So little over 100 years ago, pathologists actually thought about subtyping patients for basically the purpose of treating them in a more targeted and better way. So Broders worked at the Mayo Clinic, and he basically looked at squamous carcinoma in the lip, and he thought, oh, if I actually study the patients and I subdivide them into 4 groups, just based on morphological factors, they actually have different tumor biology and different causes of disease. So Paul Holzmann in Berlin, he worked with Virchow, who's often quoted as the founder of modern pathology, was sort of doing very similar work, but largely ignored. However, other people, like for instance Dukes, built on the work of both of them, and he looked at 5-year outcomes of low-grade renal carcinomas, and he also said if I basically introduce grades, I can actually manage the patient population better. So these patients were, these grading systems were based on morphological factors. And of course, to implement them and to basically disseminate them widely, they need to be basically quite robust and well-defined. So, as I said, you know, their implementation largely hinges on the dissemination. Now, when we sort of say what we know about disease today, those grading systems don't really capture what we know about disease progression, and importantly, they don't really mimic or capture at all what we know about molecular understanding of disease. Of course, grading systems are based on consensus. You know, groups of people have sort of refined grading systems over the years. And the outcome, for instance, your Gleason grade for a prostate biopsy is shared in a tumor board or in a multidisciplinary team meeting. So they get communicated between clinicians. So even the grading systems that are in use today have actually been validated on relatively small numbers. You saw them on the last slide, 500, 600 sets of patients. And what we should also bear in mind that their diagnostic reporting is not all that well standardized across the world or even across different regions. So I want to give you sort of 2 concrete examples how AI applied to digital pathology can sort of move beyond that and sort of show some real impact. We're doing this on a substantial digitization effort we have done in Oxford, which was part of a larger national effort. So mid-pandemic, in the summer of 2022, we had the cellular pathology in Oxford fully digitized and accredited by UCAS, which is a requirement in the UK. So today, we basically digitize between 1,300 and 1,500 slides every day, and 20 consultants can report out digitally wherever they are in the world, whether they're working from home or in the clinic. So one of the first examples I want to discuss is in the context of colorectal cancer. And we know that there is a subgroup of colorectal cancer patients that do not respond well to radiotherapy. Actually, they don't have a complete response, and it's basically we have to find alternative ways of treating those patients. So this is work from a student in the lab. It's like Ruby Wood basically asked the question, can we use morphological features and combine this with sort of our molecular understanding of disease and predict, in some sense, response to treatment? So we know that the spatial organization of the cancer is important. We know that there are interactions between the epithelium and other compartments. We also know that these have prognostic relevance. We're building here on the molecular classification of work Guinea and others have published in Nature Medicine quite a while ago on the common molecular subtypes. So this is a biological, basically, categorization of disease. I'm going to comment on that in a minute. And what we know is that there's one molecular subtype, CMS4, which has some features of stromal invasion, and there is some evidence that these patients have worse relapse-free survival. So the question is now, can we combine this? Can we combine this into sort of one model? So the opportunity we've taken here is that we previously developed a predictor of CMS subtypes, so we can basically we don't really need to rely on RNA sequencing. We essentially just take the image as a standard H&E image, what you see there. We outline the tumor area, and then we can basically use a deep learned model to predict now on a fairly fine-grained basis locally what is the CMS. So CMS basically, as I said before, it differentiates between 4 different subtypes, and these are not just individual pathways, these are sort of broader classes of biology, and I think this is also quite useful because these are biologies that actually change the tissue architecture or the microanatomy in a certain way that we can pick it up from histology samples. So what we did, we worked in a relatively small cohort that was collected in the UK and Scotland. So we have what you see here. We have about 360 digitized slides, and we basically know the CMS status of those patients. And now I want to basically outline you a little bit how we work on a predictive model to look at response to therapy. So given the size of histology images and the requirements of GPU hardware, you have to basically break them up into tiles. But then what we do is we basically using a kind of segmentation which is called superpixel to look at groups of cells that are basically similar in morphology. And we construct basically a graph out of all of this, and we sort of see what is actually sort of almost like the community of cells that are more or less interacting. At the same time, we're using self-supervised training on visual transformers to sort of learn a more or less general feature representation of that data. And then we combine this with a graph neural network that now predicts 3 different things. It predicts response to radiotherapy, it predicts the status of CMS4, and it predicts something about the spatial organization of the tissue. So we want to know where are the epithelial boundaries and where are the tumors. So by basically asking a model to predict these 3 different things, we sort of try to make the prediction problem a little bit harder, and we're also trying to sort of to constrain the problem in a little bit more targeted way, and I think this was also discussed in some of the talks this morning. So this is the loss function. The loss function is essentially combines something around the 3 different branches. And what you see in the next slide, that even on the small data set, we do get a good amount of signal, and we actually can produce response to radiotherapy, prediction of CMS4, and the prediction of the epithelial boundaries with a decent AUC. Now, I'm fully aware that this is a study on a relatively small cohort. We are just trying to extend that to basically external validation cohort, and it's not easy because it's quite a hard problem. But what I want to show you is what you can actually do with this information. So if we now have a biopsy, we can produce essentially these 3 different maps at the same time. We can produce response to radiotherapy, we can produce the response of CMS4 and the epithelium. And the idea is now that this information is a little bit more accessible, and it's not just accessible to a trained pathologist. So for instance, now you can look at the activation of these nodes in a graph, and you can sort of see that high CMS4 activation actually corresponds to low activation in response to therapy, and you also see that high CMS4 activation is present in certain stromal regions. So all these basically images have been reviewed by a pathologist, And the idea I want to sort of seed now is, can we actually change the way pathologists communicate with, for instance, oncologists, but ultimately also to patients? So the therapeutic decision lies either with oncologists or with the multidisciplinary team, and it relies really on other molecular tests. However, the pathology report is one of a key finding. And imagine in the future, we can sort of visualize that information in a far more basically accessible way and go away from the sort of grading reports I was introducing initially that are inherently limited by language. We can now essentially connect morphological assessments with molecular profiles. The second example is in a subtype of blood cancers. These are myeloproliferative neoplasms. These are essentially abnormalities in the bone marrow that affect, for example, platelet-producing cells. So what you see on the left there, that's the disease categorization, and we are particularly interested in the myelofibrosis because those patients basically have adverse events and they often need a bone marrow transplant. So a while ago, there was a wide belief that with more widely deployed genomics, we actually don't really need the input on pathologists on those cases. Really, in practice, the very opposite has occurred. So while we know the driver mutations for instance, ET, PMF, and PV, there's a really large overlap between those disease subtypes. And I want to now show you a couple of algorithms we have actually developed to sort of make that morphological distinction more accurate, and I also want to outline how this can be applied to clinical trials. So when you look in the WHO guideline and you look, for instance, what is one particular grading criteria here, that's essentially the shape of megakaryocytes. So here you see 6 different examples of megakaryocytes, and they have basically fairly abstract names, so it's not the easiest classification to learn, and it's known to be purely reproducible. We have developed essentially a data-driven workflow where we segment cells, we phenotype them, we basically say, okay, there's evidence for about 9 different phenotypes, and you see those in the next image here, so you see the phenotypes on the left, and then we can superimpose those on basically a bone marrow biopsy. And we initially thought, well, this is already great help to a pathologist because you could sort of see there's really more, for instance, in cluster 7 than I would normally expect, or more in cluster 9. But what it really turned out is that if we look in the entire cohort of patients, we can, with actually relatively simple methods, just as PCA applied on these morphological fingerprints, we can basically show these disease categories in some kind of on some kind of data manifold. Yeah, so here you see, I know it's hard to see in the back, but what you see on the right, these are 2 patients at 2 different time points, and they both have progressive disease. And you see that basically the circles and the triangles are really in different parts of this disease space. Similarly, we looked at the grading of fibrosis. Here, the WHO guideline basically just prescribes 4 different grades, which really don't capture the heterogeneity of the fibrosis in the marrow. So you, for instance, see we have a method where we look at local, basically, fibrosis grades, and you see there are certain hot spots in there, and you also see that there are areas in which the bone marrow is relatively normal. And what we can do now is almost like with the same idea of looking at the statistics or the changes in the statistics, we can now track patients over time. So when you look at patient B there, You see, this is a patient that has a bone marrow transplant, and over the course of 18 months, the bone marrow actually normalizes and sort of moves into the green spectrum. That means it's actually has normal bone marrow, and this patient has essentially a decrease in the fibrosis coefficients in the way we compute it, and it's basically is in a normal state again. So the question is now, these methods are way more accurate than what pathologists can report out today, and can we include this in clinical trials? So this is the example of applying this fibrosis measurement to a relatively small clinical trial, but we looked at this retrospectively. We had about 50 bone marrows from 3 different time points. So this basically gives you an idea what these bone marrows look like. There's some which are basically, reasonably helpful or not really myelofibrotic, and then there are ones that are really myelofibrotic. So the first thing we observed is if we basically plotted this and correlated this with our understanding of our cohort in Oxford, there were really patients part of that trial that didn't really have any myelofibrotic disease. So you see here that there are, if you look basically further to the right, to the left in the graph, there are patients that should have never been part of that study because the disease target is really not appropriate for them. What you also observe is that if we look at manual grading versus the sort of AI-assisted grading, that the dynamic range of the manual grading on the top is fairly limited. It's basically only if pathologists agree on a whole step change in grade, they will basically realize this, whereas we basically look at changes in fibrosis, we can capture this fairly readily. And if you now basically look at the whole trial population, which was thought to be inconclusive, and basically at this stage, it wasn't clear whether they would be going into a phase 2 study, we could relatively easily differentiate between reduction in fibrosis as measured by we call it continuous index of fibrosis score, and the overall increase in fibrosis score. And if we basically look into these a little bit closer, you can even find other subgroups. And these other subgroups are likely correlate with disease state, basically the time of onset of disease, and where basically the fibrosis development is. So it's not that, for instance, the drug is ineffective. The drug is essentially acting differently in different disease states. So what you can also do is you can now look at the responders. You can look at the good responders. You can plot the basically information in a longitudinal sense, and you can provide a whole lot of other information that would not be accessible or not be present in basically written reports. So I hope that gives you some idea of what you can actually do in the imaging space with AI methods. You can work on prediction models, but you can also work on really making measurements that we are doing today in a categorical sense far more precise. So I hope I was able to sort of communicate. There's a lot more information in especially these types of images, but also in radiology level images that we are using today. We can make disease diagnosis a lot more accurate. We can also link basically molecular traits with morphological features, and we can essentially bridge between pathology in a way that is practiced today with all basically advances in molecular basically treatments and molecular testing that become online. And lastly, I wanted to also highlight that these kinds of techniques allow us to communicate these results in a way that it was not possible before. So we have already shared some of these visualizations with patients, and where you sort of say, look, how would you react if you see a report that not just gives you a number, that tells you how you relate to the overall disease cohort, And some patients are quite excited by this because they want to sort of know, they would like to have something quantitative, they would like something they can compare and sort of see how they basically make their own decisions on that way. Yeah, I would like to sort of thank all the people that have done the work, and I'm open for answering questions.
Thank you very much. Any question from the audience? Okay, if not, I may start. Actually, asking this, oncology is likely the field closest to the concept of precision medicine, so far, which is miles away from neuroscience, for example, both in terms of diagnostics and treatments, as you've shown very nicely. But actually, oncologists at the same time has to face with the issue of the tissue-specific genomics and epigenomic changes in biological environment, which are distant and different from those where the cancer started. Bone metastasis, for example, from lung cancer. Yeah. So how are you facing this? What is the approaches for the progression of disease in oncology that sometimes has to do much more with the progression in different organs and likely responding to rules, to biological rules which are different from those?
Yeah, I think ultimately, if you work on say, let's answer this in almost like two steps. If you are confined to pathology data, you're basically confined to a sampling bias where biopsies are being taken from. But I think what we now basically also start is correlating this, for instance, if you correlate this with radiology data, then you can sort of almost like see, say you combine pathology with radiomics signatures like radiomics, and then you could sort of see, basically metastasis is a signal and metastasis different than in basically the biopsies. But I think what's already happening today is that what we can, for instance, do, we can also look at tissue in 3D, we are, for instance, already seeing clear evidence that there are features outside of the tumor, for instance, in the stroma, that are clearly predictive. Yeah, and that's basically the AI field has sort of enabled the discovery of these, and I think that's almost like the journey we need to expand on, basically correlating this with radiology measures as well.
Congratulations. I also have a question for you. I'm working in metabolic diseases and especially in the liver. Liver fibrosis is also another place where AI will be very much welcome, because you have just a small sample of the liver, and it's very important, as you've shown, to look if there will be an improvement with treatment, and having with AI a scale of fibrosis, and not like the 4 from F0 to F4 as a pathologist is doing, is very, very important. My question is, do you think in the future, especially for large organs or whatever, there will be the possibility to link the results in biopsy in tissue with the results from radiomics, for example? Because of course, not only the patient wanted to see results, but there will be the possibility to do it more frequently, the sampling, so the possibility to link images from a tissue with imaging from MRI or another kind of...
Yeah, I think you're absolutely right, and I think the liver is a great example, because you basically have the liver, the biopsy is a very invasive test, right? And for instance, we're doing similar work in liver fibrosis, so you can almost like follow a similar paradigm there. But there you need to correlate with, some people are basically advocating MR signatures, but then we also have the whole topic of liquid biopsies, where I think where there is an opportunity of finding almost like priors at baseline that will allow us to look at liquid biopsy and, for instance, MR in perhaps a more targeted way than it is done today. Yeah, because liquid biopsies will have certain complications, and people have also already shown this. It's quite varying metabolites.
No, yes, for the liquid biopsy, yes. But, you know, for example, there are MRI, you know, magnetic resonance elastography or imaging that are also linked to fibrosis and inflammation. But of course, you know, right now the gold standard is the biopsy, so the tissue biopsy. So the possibility to link, you know, whatever you find in the tissue and whatever you can see as an image with radiology will be the future, I think, because there will be, you know, taking out the invasive of a biopsy, and also the biopsy is just a small sampling of the big organs. So AI will be definitely
absolutely, and I think the sort of pathology results will also be critical to validate some of the new MR signatures.
Thank you. Thank you very much again.
So we go to the next talk, Professor Fabio Van Dien from Department of Information Engineering in Padua. He's Professor of Computer Engineering, and the title of his talk is AI in Cancer Genomics, Challenges and Future Direction.
Okay. Hello, everybody. Thanks for the wonderful introduction. And I'm very happy to present today some of the methods that we have developed to analyze mutations in cancer. So I'm a computer engineer, and so we develop algorithms, computational techniques, and what I'm going to present today are two of the methods that we have developed to analyze somatic mutations in cancer. So somatic mutations are crucial in the development of cancers, since it's known that they are very important in the transition from the normal cell that leads to cancerous cells. Not all somatic mutations are important, though. Some of them confer selective advantage to the cell, and they're usually called the driver mutations, while most of the somatic mutations that you can find in a cancer genome are actually passenger mutations. They do not have an effect on the cell. So what we can do now, thanks to sequencing technologies, is actually take a cancer or a tumor and sequence it, and therefore obtaining, let's say, a measure of the mutations in the genome of the patient. But of course, we don't know which mutations are driver mutations and which ones instead are passenger mutations, okay? And the question that we want to answer is actually, of the many mutations that we observed, which ones are driver mutations and which ones are not? This is a difficult question to answer for many reasons, but like one of them is the fact that if you look at the numbers, there are probably a handful of driver mutations in a cancer genome, while there are hundreds up to tens of thousands of passenger somatic mutations in a cancer genome. Now, thanks to the advances in sequencing technologies, you can actually sequence a large number of patients for different tumor types, and this is the endeavor that has been taken from different projects at the international level, including the Cancer Genome Atlas and the International Cancer Genome Consortium, that provide now publicly available data for more than 20,000 patients, for which you have all the somatic mutations that have been measured in their exome or in all their genome. And so what we can do now is leverage these fairly large data sets to look for patterns of somatic mutations in order to try to identify important mutations in cancer and obtain significant insights in the development of the disease. So there are several approaches that have been developed to this end. And some of them, for example, look for recurrent mutations or recurrently mutated genes. So you look at one gene at a time, and you essentially look how frequently it is mutated. And if the gene is mutated in a large number of patients, then you say it's a significant gene, so it contains driver mutations. Or you can look for different types of patterns, like clusters of mutations, so mutations that appear in specific positions and accumulate in specific positions of genes, or more complex patterns like mutational signatures. So these are good approaches, but they all share the drawback that they only work well for frequent mutations. So you can really detect only driver mutations that appear in a large fraction of the patients. However, if you look at the data, actually there are few genes that are mutated with high frequency in a cancer cohort, while most of the genes are mutated at a much lower frequency. And if you look at the numbers, most of the driver mutations, or many of the driver mutations, will also appear in this long tail of low-frequency mutated genes. And actually, this relates to the observation that is the so-called intertumor heterogeneity. That is, if you look at two tumors, even from the same type, they don't share the same complement of somatic mutations, okay? And in fact, if you look at the data, so like this is data from the Cancer Genome Atlas on ovarian cancer, and if you look at the patients on the rows, here you have that mutations are those blue dots that you can observe, and you can see that the data is very sparse, and there are probably no two lines that are identical in this simple representation. So finding patterns in this type of data is very complicated. However, artificial intelligence can help us to identify significant mutation patterns and reveal interesting insight about the disease. So now I'm going to present two approaches that we have worked on. And the first one actually looks at mutations in the context of networks. So it's well known that you can look at the cell as a complex system with interacting elements, molecules, okay? And it is actually the interaction of these molecules that perform some function. So part of this complex circuitry is performing a function, and by abstracting a little bit, we can think that for each of these so-called hallmarks of cancer, the characteristics that must be acquired by cells in order to become cancerous cells, there will be, for each of these hallmarks, like one subnetwork or multiple subnetworks related to such hallmarks. And for this reason, if we can alter the function, alter the subnetwork by simply mutating or altering any of the genes in the subnetwork, we can observe then genes that have the same effect, altering a given function, but with very different frequency of mutation. And so instead of looking then for single genes that have a lot of mutations or that are frequently mutated, what we should really be looking for are subnetworks of this large network that are significantly mutated. So this idea actually, it has been around for a long time, and there are several methods to do it, and many methods actually look for known pathways. They only consider subnetworks for which the actual function is known. And what you can do in this case is then generalize the methods that look at each gene in isolation by looking at each subnetwork in isolation, and then assess whether for a given subnetwork the number of mutations that you observed is higher than what you would expect. And if this number is high enough, then you would say, well, this known pathway is significantly mutated in my cancer cohort. Okay, so this approach sometimes works, but it has several problems, including the fact that not all pathways are known, so we don't know all the pathways that perform functions. These pathways are actually not distinct entities. They do interact with the so-called crosstalk between pathways. And sometimes these pathways in the databases that you can use are actually huge, and instead only a small part of the pathway may be important for cancer. So what we really want to do then is not to rely on this well-detailed knowledge that is important but doesn't give us the complete picture of what are the mutations that are important in cancer, but we want to find subnetworks of a large interaction network that is significantly mutated without restricting only to known pathways. Okay, so this problem is very complex, is very challenging for several reasons, including the fact that there are many candidate subnetworks that you should evaluate. So even if you restrict to small subnetworks with at most 6 genes, there are more than 10 to the 20 candidates they should evaluate somehow. And this poses a severe challenge both computationally and statistically. So what we have done a while ago is to develop the first method, HATNet2, that actually takes in input mutations, or better, alterations, both single nucleotide variants and copy number aberrations, and deriving a score for each of the genes, it then applies a diffusion process on the large interaction network, and using this diffusion process, at the end, it identifies a small number of subnetworks that are significantly mutated. And you can find the details in the paper, but one of the important things for this type of approaches is that we can rigorously assess the significance of the findings that we report. And just to give you an idea of one of the analyses that we have done is we have used HATNet2 to analyze data, somatic mutation data from more than 3,000 samples from 12 different cancer types in a pan-cancer analysis. And by applying our algorithm, what we find are 16 subnetworks that contain 147 genes in total out of the 20,000 genes that were given in input. And this is a plot that shows, and hopefully is visible from the back, what are these subnetworks or a representation for these subnetworks. And what is interesting here is that some of the subnetworks actually recapitulate some of the knowledge about cancer and correspond to a very well-known mechanism in cancer, like p53 signaling, while other subnetworks are not as well characterized. And this is done fully automatically without actually an intervention from the user. Okay, so just to give you an idea of the type of results that we find, an interesting one was about cohesin complex, where we identified this group of 7 genes, where one of them is mutated at kind of a fairly high frequency, while the other ones are mutated much lower frequency. But overall, the subnetwork is significant as assessed by our technique. Okay, and later on, there were multiple reports that actually cohesin is a very important mechanism in cancer and for different cancer types. So this is a method that we developed a while ago, and we have more recently generalized this method to also look for subnetworks that are associated with different phenotypes. It could be survival time, for example, or drug sensitivity data, and we have also extended it to consider mutational signatures or to look for drug response. Okay, so this was a part about inter-tumor heterogeneity. And now I'm going to switch gears and talk about methods that we have developed to tackle a different type of heterogeneity, that is the so-called intratumor heterogeneity. And intratumor heterogeneity is the observation that if you look at a tumor, is actually composed very frequently not just by a population of cells that share the same somatic mutations, but actually there are different subpopulations that are of course related, but they have different somatic mutations that you can observe. And this is due to the fact that if we look at cancer as an evolutionary process where you start from a founder cell, then the driver mutations that accumulate can actually lead to different subpopulations characterized by different important mutations in the tumor that you observe at the end. Now, this nice picture about the evolution of cancer can actually represented mathematically and formally. And one way to do it is actually to represent it with a graph or a simple network, as the one that you can see here on the right, where you have on the top the normal cell, and then you have the different steps that led to the observed tumor that you have, with the accumulation of mutations that are represented here by the different colors and the different symbols. Now, obtaining these three from a tumor is not easy, and you can do it by using, again, DNA sequencing data. You can try to do it with bulk sequencing, and it's very difficult. You can do it with multi-region bulk sequencing, and you get better results, or the best thing to do would be to do single-cell DNA sequencing. But at the end, with different algorithms, you can try to infer the tree that describes the evolution of the tumor. Okay, so this is, again, a difficult problem, and many, many methods have been developed in recent years to try to infer the evolutionary tree for a tumor given the sequencing of the DNA of the tumor, starting from different types of data. Now, what we have been interested in instead is not reconstructing a tumor tree or the evolution of a tumor by itself, but actually try to find patterns in the trees that describe multiple patients. So you can think that you have your patients, and by DNA sequencing and using algorithm, you can infer a tree that describes the evolution of each tree, the evolution of the tumor for each patient. And then what we want to do is to identify conserved progression patterns of alterations in this collection of evolutionary trees for tumors. Okay? And This may look like a simple problem, but actually is very difficult because the patterns we are looking at are not mutations that must be subsequent in the tree. So we don't need them to appear one immediately after the other. But actually, they could appear, of course, one after the other, but with many mutations in between. And this really complicates the computation for the identification of these patterns. Okay? So we have developed recently a method called the Mastro that actually finds conserved trajectories in tumor trees. And it can identify any type of trajectory, so without restricting, for example, to simple linear chains as previous methods. And it also provides a way to assess the statistical significance of the output that it provides. Now, to give you an idea of the type of results that we can obtain with the Mastro, in the paper we analyzed among different data sets, one on 123 patients of AML, for which we had data from single-cell panel sequencing for 31 genes, and observing in total 543 single nucleotide variants. Now, using this data and using previous methods, we were able to infer a single tree for each of these 123 patients. And then we applied our method Mastro, and what we obtained in output were 40 significant trajectories that are then summarized in the plot that you can see here. So actually, these 40 trajectories can be clustered in 4 different types of trajectories. For example, the one on the left describes the evolution starting from the normal cell and then accumulating a mutation in the epigenetic factor DNMT3A, followed by mutation in NPM1, and then you observe mutations in 3 genes that are known to be important for AML, FLT3 and RAS and KRAS. But the interesting observation that we obtain automatically is that these 3 mutations appear in 3 different clones in these patients. Okay? So this is an observation of mutual exclusivity of mutations inside the single tumors that you have in the patient. So you do accumulate these mutations, but in different clones. Now, the other modalities are related, and for example, the second one relates to the fact that starting from the normal cell, you could have a mutation either in IDH1 or IDH2, and then this is followed by mutation in NPM1 or in FLT3, and there are also the other types of ways to progress to AML that you observe for the other 2 panels. Now, these 4 types of evolution for AML actually correspond for the first 3 to non-progression patterns that were reported in the paper that made the data available and also in the subsequent papers or in different ones, while the last one has not been previously reported and it's well supported actually from the data, and it may correspond to a different way in which AML arises for some cohort that have not been previously reported. So it may correspond to a novel discovery that we made by using our method. Now, just to conclude, I really want to briefly mention 2 directions that we are just started really to explore in terms of using artificial intelligence to analyze somatic mutations in cancer. So the first one tried to tackle the problem that while we have fairly large data sets, let's say with thousands of samples, in many cases, these data may not be enough for some of the deep learning techniques that provide very useful tools to predict, to make predictions about cancer patients. Okay? So what we want to do is try to enrich this type of data, for example, by combining all exome data and also gene panels data that very often are easier to obtain because of cost, for example, and also they are easier to collect from different studies. And so one thing that we are doing right now is to use graph neural networks to combine them by essentially trying to solve the problem of generalization to out-of-distribution data, so that by learning a model, for example, with exome data, we may be able to use the same model for gene panel data, so that it works also we don't have all the information about the mutations. And this model can be used, for example, to predict the response to some therapy or to predict the course for the disease of a patient. And then on a different line, we have been developing methods that try to identify causal relations to cancer phenotypes, so making some assumptions, of course, about somatic mutations and their relations, as it is always needed when you talk about causality. And we have been developing a method that looks for causal rules so that we identify causal relations between somatic mutations and different cancer subtypes. In terms of future direction that I think are essentially still open for the analysis of somatic mutations in cancer is the fact that, as we've heard various times today, somatic mutations or mutations in general or the DNA in general is not everything. Gene expression is important, DNA methylation is important, the environment is important, and the way to integrate all these different types of data is still an open challenge in the field. And then we are interested also in integrating tumor trajectories, so what we can characterize with Mastro, with subnetworks and pathways, what we can characterize with HATNet. So obtaining essentially a network of networks that can really describe both the evolution for different patients, but also at the level of the single patient. And finally, we are interested in incorporating data about the spatial evolution of cancer, since, of course, the way the cancer develops is constrained also by the spatial structure of the tissue, and is something we are really interested in and that we are exploring for the future. Okay, so to conclude, I hope that the two methods that I presented, HATNet2 and Mastro, that look for patterns of somatic mutations in cancer, have shown that it is very important to use artificial intelligence tools to identify important patterns of mutations in cancer. And all the software is available at the link that you can see here. And I just want to acknowledge all the members and the students and the personnel that has worked on the different projects, and also the collaborators that really have made all these works possible. So thank you, and we'll be happy to answer questions.
Yes, thank you. We have some questions from the audience.
Thank you very much. Again, Raffaella Bisson. Nice to see you again, Fabio.
Nice to see you too, again.
One question. What about photoacoustic imaging? It is very powerful because we know now that cells communicate through light, photons, sound, phonic signals, biochemical signals. So we have these three signatures, and we can investigate on soft tissues with these three kinds of signatures. Are you thinking to use also these opportunities to trace these signatures? Because tracing these signatures, we can follow the evolution just before the pathology, just while changing the cells. So we can prevent the final disease, the severity, I suppose.
Yeah, so that's a great question. And of course, integrating as much information as possible would make the method and the patterns that we can identify more useful. I have to say that sometimes one of the problems is the data availability. So we haven't been able to get good data sets for some of the data types that we are interested in. But of course, it's an interesting direction, how to try to incorporate all the possible information that we have so that we can try to predict what will happen as soon as possible, essentially. And it's tough. There are some papers that actually show that there may be some intrinsic limits, in the sense that there may be so many different pathways that the disease could take for some cancer types, that then it may be difficult to predict reliably which direction it will go. But the more data you have, probably the more opportunities you have to find a good way to characterize which direction is going to follow. Thank you.
Thank you. A very quick question. Actually, you showed us some data which remain within the paradigm genotype-phenotype. Okay, the phenotype is a tissue phenotype or cell phenotype, but essentially it's a phenotype. And this paradigm actually turned to be not that effective in medicine as we hope to be, at least actually most of the cases. So here, as you've shown, as you will be you're dealing with changes which might be driven by other factors. So what about scaling up, including transcriptomics up to translatomics, but that could increase very likely the probability to?
Yeah, so that's a great point, and that's essentially what we want to do by integrating also the other type of data. So one thing that I like about the DNA is that it's fairly stable. It's not stable as, you know, since the evolution that you have in tumor, but it's a fairly stable, and you can measure it fairly accurately, while other types of data are more, you know, they are more dynamic. And of course, that provides a lot of information, but also from the point of view of then using it maybe in the clinic, it may be a bit more complex. And I think it's essentially an opportunity, the one that is in front of us, of really integrating all the different data types to get the best out of all of them.
On this line, I have also a question for you. Do you think it will be possible with your type of approach to integrate also the imaging of the environment of the cancer? I mean, because of course the cancer could be different according to where it's located. It could be in the adipose tissue because of the other organs. So I think not only the imaging of the cancer, but also the environment that is the tissue around the cancer will be important.
Yeah, that's definitely a very interesting direction, and I think there are opportunities to integrate the data that you have at the molecular level with the imaging about both the tumor and the environment. How to do it, I'm not sure how to do it yet, but that's you know, it's something that we can work on. Or I should say there are some straightforward ways to do it that I don't think will work very well. But then that's, you know, what we do is to develop methods, and so we are very interested in going in that direction.
Okay, thank you very much. Thank you very much,
Thank
Fabio.
you.
And we have to move on. And I'm pleased to invite Professor Davide Risso. He's Associate Professor of Statistics at the Department of Statistical Science at the University of Padua. Will be giving a talk on statistical modeling of spatial transcriptomics data. Thank you. It's 12 minutes.
Thank you. I guess my slides will start. All right. Yeah, thank you so much for the invitation. I'm really happy to be part of this really great event. So I'm going to be talking about spatial transcriptomics data in these 15 minutes. But I also added a subtitle to my talk because I'm in a Department of Statistics, and I wanted to also give you, I guess, my view of what is the role of statistics in AI. So I guess I'm really lucky because Dr. Richter did a great job at making me skip my introductory slide about computational pathology. Here is essentially like one other example of what he showed beautifully before, sort of how we can use this like digitized whole slide images to essentially extract features that are able to be used as prognostic risk scores, for example, with prognostic or diagnostic features in mind. But maybe what I can spend a little bit of time is the other side of the coin when we talk about spatial transcriptomics, which is gene expression. And we, Fabio just described very beautifully how we can use DNA sequencing as a precision medicine tool. Here we're using sort of like the next level in omics, which is transcriptomics. And I just want to give this one example that appeared a while ago in the New England Journal of Medicine, where they focused on a essentially transcriptomic test for the diagnosis of thyroid cancer. And approximately 15 to 30% of thyroid nodules that are evaluated after biopsy by pathologists they're not clearly either benign or malignant, and typically patients get sort of referred to diagnostic surgery just in case. And this group developed a 167 gene panel that essentially works as a test, as a diagnostic test. And out of this number of interdeterminate nodules that they analyzed, 85 were malignant, and they were able to correctly identify 78 out of these 85 nodules as suspicious with a pretty good sensitivity. So in addition to sort of like pathology, we have this additional tool, gene expression, that is indeed can be used as diagnostic or prognostic tool. And I guess the second piece of the puzzle is that, of course, these two data types are not independent, right? We can show, and this is a nice example of that by Barbara Engelhardt group at Princeton, which they show using the TCGA data that Fabio just described, how you can use morphology to predict gene expression. So they use a convolutional neural network to extract some sort of features from latent factors from imaging data from H&E staining, and they were able to sort of associate these factors with gene expression and statistically enrich for interesting gene ontology terms. So essentially the missing piece now is a technology that allows to measure both in the same tissue. And that technology is now a reality. It's called spatial transcriptomics. And essentially by spatial transcriptomics, what we mean is the attempt to quantify a large number of the mRNA from a large number of genes, ideally the whole transcriptome, within the spatial context of the tissue and the cell. And so in terms of protocols and data types, spatial transcriptomics technologies can be sort of like broadly defined in two different categories. One of them is what people call next generation sequencing-based or spot-based techniques that are sort of, by definition, they measure in an unbiased way the whole transcriptome, but they're not at the cellular resolution. They're typically at a supracellular resolution. On the other hand, you have in situ techniques that typically rely on a predetermined set of probes. So you're biasing yourself for measuring only certain genes, typically a few hundreds up to a few thousand genes, but then you have subcellular resolution. And so you can really see exactly the position of each molecule of each mRNA within the tissue, and you're basically just limited by the optical resolution of your microscope. So at the end of the day, you have this sort of numerical matrix where you have your genes in the rows and your spots or cells in the columns, and along with the XY coordinates of your cells. But you also have the original images at your disposal. And so the hope is that you can integrate all of these data to kind of get a better view of the sample. So now that I sort of give you an overview of the data, I wanted to sort of get into this like subtitle that I added. So what is the role of statistics in all of this? And very recently there was this statement by the American Statistical Association about the role of statistics in artificial intelligence. And I don't have time to go into the details, but I think it's a one-page document, and it's a nice read if you have time to dig it up later on. But there's sort of like three pillars or three basic principles that I in some sense, I would like would be the sort of take-home message from my talk are interpretability, reproducibility, and uncertainty quantifications. These are sort of like the three pillars that statistics and inferential analysis can sort of bring to the table of AI. And so my hope now is to give you kind of like three research snapshots that have been going on in my group. They're sort of like are sort of examples of these three sort of pillars. So the first one, interpretability. We have been developing this model that we call three-phase. It's a three-factorization algorithm for spatial expression. And the idea here is that we have our data matrix, the one I was showing you before. You can imagine sort of like genes on the rows and cells on the column. And the idea is that we want to sort of factorize this matrix in three components. This is essentially similar to non-negative matrix factorizations for those of you that know that. But instead of two components, we're using three components. And that allows us to essentially simultaneously cluster gene sort of rows and columns of that matrix, which is essentially a gene-cell co-cluster. And this is my only, I guess, my most technical slide, but I just wanted to put it here to show that this is actually a quite simple model. Of course, we could do like kind of some kind of convolutional neural network or graph neural network or whatever, but it turns out that this sort of linear sort of loss function still works well for this type of problem. And the only sort of complication is that we want to include sort of the information, we want to inform the model on the spatial dependencies of the cells in space. So instead of a straightforward sort of matrix factorization, we had this sort of Cholesky factorization of an isotropic kernel, which is just complicated words to say that we inform the model that two cells that are close to each other should be more correlated. So this is an example data set in which we applied this model. It's from this 10x Genomics Visium technology, in which you kind of see on the right-hand side of the slide these circles. These are the spots in which we are measuring gene expression across the tissue. This is a prostate invasive carcinoma. And on the left-hand side, you see this H&E image that has been manually annotated by an expert pathologist. And you can see the blue area on the top is the carcinoma, and essentially in the bottom part we have stroma. And here is essentially the results of our model that is completely unsupervised. So I'm keeping on the bottom left side the manual annotation by the pathologist, but the model was not informed by that. And what we can get out of these models sort of like gene and spot clusters. And for example, cluster 1 is essentially marking fibromuscular tissues. And we get genes in this cluster that are enriched for muscle proteins and muscle processes. Similarly, for cluster 2, we essentially mark the invasive carcinoma, and we're able to identify gene signatures that have been associated in the literature with invasive carcinoma. And then, you know, I don't have time to go through all the clusters, but for example, just to show you that we are also able to find a sort of rare cell population, like for example, there's a nerve at the bottom of the tissue, and we're able to actually mark that with a specific cluster of genes. Okay, so this is sort of like moving towards this idea of personalized medicine, right? We are training a model on data from a single patient, and we're able to sort of maybe help the pathologist to one more tool to look at this specimen from one patient and help with a better diagnostic or prognostic signature. But there's sort of this tension in my view between personalized medicine and effectiveness of a treatment, right, or robustness of a treatment. We want to know if the treatment is effective in a single patient, an individual patient, but we also want to test the effectiveness of a treatment in a population, right? And so if we don't do that, we risk overfitting, and we all know this sort of like bias-variance trade-off. So the second idea that I sort of want to sort of highlight, and this goes towards this reproducibility as the second sort of like topic that I mentioned before, is can we actually reuse cancer signatures that have been developed and studied in large cohort studies to learn something about new samples, perhaps at an increased maybe spatial resolution. And just to give you an example, there's this paper that analyzed gene expression from almost 5,000 patients with breast cancer. In the end, they learned a 51 gene epithelial to mesenchymal transition signature, and they validated as a predictor for late disease recurrence. And this is just one example. We collected many of these signatures from 150 papers. And then we can apply it again to this kind of like spatial transcriptomic samples. This is another example, again, 10x Genomics Visium technology. We have a breast cancer sample in this case, again, annotated by an expert pathologist. And here, if you focus on panel E, this is the EMT signature that I just described. And you can, again, this model is completely unsupervised. It doesn't see the manual annotation by the pathologist, but it's pretty obvious that it's doing a superb job at sort of separating tumor from stroma, right? And which which you can see from the manual annotation on the top left corner. And even if you look only at the tumor spots like we're doing in panel F, you can see that you can sort of see sort of like hot tumor versus cold tumor, which is, I guess, also seen in panel D, where we look at hypoxia signatures. So this is, I think it's a very good trade-off between personalized medicine, but kind of like robust description of signature and testing of signatures. So the last word that I mentioned is this idea of uncertainty quantification. And here I'm just very quickly showing you the last snapshot here, which is related to cell typing, which is a critical step in this kind of analysis, because the first thing we want to do is to assign labels to cells, so give a cell type label. And state-of-the-art models, they not only give a point prediction, but they also give a sort of like probabilities of predictions. And you can see here, for example, these are 3 rows of a 100,000 row matrix, in which you see that the first cell is quite sure, the model, that it's as smooth cell muscles. For the other cells, there's a lot more uncertainty. And I don't have time to go into the details, but basically we have a way to inform these kind of models with what we know from the cell ontology about the relationship between cells to sort of give this sort of dynamic resolution of cell typing for this type of data. So I just end here with thanking my group members and collaborators, and I'll just give you a couple of references, and thank you for your attention.
Thank you very much. So there is time for one quick question. Here is the question there of a friend. Short question.
Very much.
Short
very
answer.
short. Can we consider as personalized medicine also immunotherapy? Because considering the cellular ground, and the immunity system is very important. And so instead of considering only drugs, we can consider to enhance the immunity system. Is it right? Thank you.
Right. Yeah, for sure. I mean, I didn't, these data, I'm not looking at therapy right now, right? This is just looking at sort of, I don't have like a before and after treatment type of data yet. But yeah, you're completely right. The good, I guess, feature of this kind of data is that you're not only focusing on the tumor, but you're looking at the tumor microenvironment, right? And so definitely the immune system plays an important role, and definitely immune therapy should be something to look into.
Thank you. It's a fascinating new technique, but we have to move on. Thank you very much again. And please, Emma.
Thank you. And now we move to the last talk of this session, Dr. Mario Luca Morieri, that is a clinical investigator of the University Hospital in Padova, and the title of the talk is Unmet Clinical Need, the Role of AI and Clinical Decision Supporting System.
Thank you. So it's a great pleasure to be here and be part of this exciting meeting. These are my conflicts of interest. No one of these is directly related to this talk. And today in this talk, I will focus on 2 unmet clinical needs. The first one is the inadequate prescription of statins for cardiovascular prevention, and the second one is the low screening rate for diabetic retinopathy. And what I will do is show you some example on how, I'm sorry for this problem with the slides, it was not supposed to happen. Anyway, what I will show you is how clinical decision supporting system and artificial intelligence are expected to provide benefit to solve the first unmet clinical need, statin prescription, and how this is already applied at our clinics in Padova to solve the second problem, the screening of diabetic retinopathy. So we know very well that type 2 diabetes patients with type 2 diabetes have a higher risk of cardiovascular disease as compared to the general population, and this is keep over years. And this problem becomes a major problem if we consider that type 2 diabetes prevalence is increasing worldwide consistently. However, the good news is that we have effective treatment to reduce cardiovascular risk, and one of these is reducing LDL cholesterol. LDL cholesterol has a causal role in cardiovascular disease. This is well established with genetic studies, epidemiological studies, and a randomized clinical trial showing that if we reduce LDL cholesterol, we achieve a proportional reduction in the risk of cardiovascular disease. And we have effective treatment. We have different types of treatments, with statins being the first-line treatment, highly recommended by all cardiovascular preventive guidelines. However, real-world data are not so good. As we showed in this study, multi-center study in Italy, on over 100,000 patients with type 2 diabetes, the prevalence of statin prescription among patients that are eligible to this treatment is very low, around 60%. And if we focus on those on achieving the LDL cholesterol targets, the proportion is even lower. Most importantly, this translates in unmet clinical need with a huge missed opportunity for cardiovascular prevention. Indeed, what we did, we estimate that there was, with the lack of appropriate treatment, we are wasting our opportunity to reduce cardiovascular risk of patients. For example, if we treat 100,000 of these patients with appropriate lipid-lowering treatment, we would be able to reduce cardiovascular events by 1/3. And this would save a lot of cardiovascular events in this patient. Again, I'm sorry for this problem with the slides. When I checked this in the slide, the room was not so as this. Anyway, the problem here is not local, it's a global shared problem, and this underlines that there is a systematic problems in the healthcare systems. There are several reasons for this, and one of the most common culprit is considered the patient adherence, so the patient side of the problem. However, what we did as other authors was to show that even among subjects and patients with high adherence to treatment, there is a low achievement of LDL cholesterol target. For example, these are data from 1 million subjects in the Veneto region, and when we selected patients with or without type 2 diabetes with high cardiovascular risk, because they were on secondary cardiovascular prevention, and with high adherence to treatment, again, the achievement of LDL cholesterol was very low. So we moved to the physician side of the problem, and in these other studies, we conducted a survey that allowed us to identify how physicians often misclassify the risk of patients. So this is a major problem that leads to a low achievement of LDL cholesterol target and an inadequate treatment option suggested by physicians. This suggests, therefore, that there is a need to better support physicians to avoid clinical inertia. So far, I've shown you how electronic medical records can help to identify gaps and unmet clinical needs. However, these are very well positioned also to be transformed in a tool to overcome these unmet clinical needs and implement changes. And this is integrating clinical decision supporting system and artificial intelligence. I will show, and for example, one of the questions is, can we use clinical decision supporting system to improve the prescription of statins? This was elegantly shown in these studies conducted at Penn University in USA, where the authors randomized 28 different primary care practice clinics to 4 types of intervention. It was user care, and then there was nudges to patients that were intended as text messages sent to patients before appointment saying, do you want to receive information about your health, yes or no, maybe you need a statins, you might need a statins, and you want to discuss this with your physician. This type of nudges to help patients' adherence. The nudges to physician had 2 components. One was integrated in the electronic health records, showing which was the cardiovascular risk of the patient and which is the guidelines recommended treatment for that patient. That physician could take the decision on its own, of course, on whether to prescribe or not the treatment. And the other component of these nudges were a monthly peer comparison that each physician received, saying if the prescription rate of statin was above or below or equal to the median of the peer in the same clinic. And then there was a group that combined the nudges to clinician and patients. The study showed this result, showing that while all groups had a tendency to increase prescription in statins, there was a very significant increase in nudges to clinicians with or without the nudges also to patients. And these proof of concept studies highlight two important concepts. First is that it is possible to use clinical decision supporting system to improve statin prescription. And the second one is that as compared to previous studies with similar design that failed to achieve this result, allow to identify key important elements such as the timing for the clinical decision supporting systems, the timing of the alert is very important, as well as the type of information that is provided to the physician, such as this is the recommended treatment because for this reason, for example, because the patient is a high cardiovascular risk. Nudges to patients were not so effective, and more probably this highlights the need to identify which is the most effective type and system of communication. As we heard a lot today, we need, of course, to have good quality of data. And ideally, the electronic medical records would like to have structured data with everything coded like with a 0-1, like yes, no, or with categorical variable. However, what happens in the real life is that most of information in electronic medical records are taken by free text nodes. And here is a situation where AI can really help a lot in converting a structured data to more usable information. What we did in collaboration with the Department of Informatic Engineering in Padova with some great colleagues, such as Alessandro Guazzo, Enrico Longato, Barbara Di Camillo, was to evaluate if we could identify cardiovascular hospitalization from a structure free from text of patient routine visit with deep learning-based natural language processing. We used our database to evaluate whether using free text visit we were able to identify cardiovascular hospitalization occurred in different time windows, 6 months, 12 months, 24 months, and any time before each visit. And we leverage our large database with several information on patients, collecting more than 78,000 routine visits and more than 2,000 cardiovascular events. The dataset was divided in training, validation, and test set. The engineer developed 4 different neural networks with different thresholds, optimized independently, one for each time window, and we allowed some different level of uncertainty in the response, meaning that the presence of cardiovascular events could be yes or no, or allowing also some like yes, no, or uncertain answer. We use different metrics to evaluate the performance of these scores, and overall they were consistent and showed that when we evaluate the infinite time window or the 24 time window, there was a good discrimination of cardiovascular events using this model. The discrimination was much lower for shorter time visits, and this makes sense because at our clinics, patients are seen on average once a year. So if you get a time windows close to that time or shorter than that, of course, it's easy to lose information. Anyway, within the limitation of retrospective studies, single center studies, again, these studies shows how we can transform free text information into structured data that could be used to inform electronic medical records. Another studies that was conducted by Gobel et al. Showed a similar approach to identify, for example, adverse drug reaction that is usually collected again as free text notes instead of in a structured way, and this is very important because this might allow us to identify those patients that are not treated with statin, for example, for a good reason, instead of those patients that have potential clinical inertia that should be addressed. In the last 2 minutes, I would like to discuss to show you how we are already implementing AI-based diabetic retinopathy screening at our clinics, and how this is already helping me and my colleagues in this context. So diabetic retinopathy is one of the major complications of diabetes. It's essential also for cardiovascular risk prediction of patients. However, real-world evidence shows that it is underreported and there is an inadequate screening of diabetic retinopathy. And the reason why it is so important to screen for diabetic retinopathy is because if you identify a moderate retinopathy or more severe retinopathy, these are considered a sight-threatening condition, and this requires a prior high specialist visit. In this field, AI applications have been studied since long time and with great results. We have recently implemented an AI-based algorithm nested within our electronic medical records that has been developed and tested to filter out subjects that needed to be referred to an ophthalmologist. Basically, this is based on a fast and full automated camera that doesn't need dilation, mydriasis, so it is very comfortable and it shortens some time that could be done, and it is done by nurse. And this is what it is linked to the clinical medical record. This is the output that you get from this result with the negative screening. The diabetologist can decide if he or she wants to evaluate the images and then decide to refer the patient to the eye specialist or not. The good news is that beyond the development studies that has allowed these to be classified as medical devices approved by Europe regulation. Also recently an independent studies has validated this approach, showing that as compared to ophthalmologist evaluation, there is a high level of sensitivity, 100% of sensitivity and 80% of specificity of referable diabetic retinopathy. For the epidemiologist in the room, 80% specificity might seem not so high for a screening tool, but we should consider that in alternative, what we are used to do is to send anyone to the eye specialist to examine the retina of this patient. So these are actually very good numbers, and what we need here is this high sensitivity. In conclusion, again, sorry for this problem with the slide, but I hope that I showed you how electronic medical records implemented with the clinical decision supporting system and AI are well positioned not only to develop new algorithm and new treatment, but also to close the gaps between real-world data and guidelines recommendation. This is already applied for some, in some cases, and actually much more will enter as such of this algorithm in the future, but this will require, of course, further validation. There are some future challenges, of course. First of all, the training to correctly use artificial intelligence and clinical decision supporting systems. This is very important to train healthcare providers, but also patients to deal with type of the information that are derived from AI. And another point, as we highlighted in the international consensus for precision diabetes medicine, but that can be applied to any cardiometabolic disease, is that we need appropriate report of studies, transparency, and accessibility to facilitate and accelerate equitable clinical implementation of precision medicine. And equity, I think, is very important here. We should think that we are facing a lot of inequity in healthcare systems and especially in these days. We know, we have heard also this morning about war, and we know that there are a lot of inequity in the world, so we need to develop methods that can be applied to everyone, like both sides of the world need
Mario,
to
please,
apply
go to the conclusion, please. The time is over. Thank
50 minutes, yeah. And so this is very important because as physician, if you want to apply this to we need to apply this to anyone, and so otherwise we will fail and we will just increase inequity. Thank you.
So we have time for just a very brief question. If nobody has one, I just have a short question for you. We heard this morning from Bjorn that AI in commercial things like the red bike is choosing, which is the best model. Do you think in the near future the doctors will have something like that for drug suggesting drug treatment? For example, for diabetes, there are drugs now that can cover cardiovascular disease, glycemic control, and so on. And do you think that will suggest the doctor?
Yeah, I think we have two components here. One is using AI to identify the best treatment to patient, and the second is AI and clinical decision supporting system to just apply what we already know to be effective that we are not implementing. So yes, in the future I see that we will have both this type of information.
Thank you very much.
Thank you. So thank you to all the speakers for the great talks and all of you for attending this session. Thank you very much.
So just to give you information, so we have a break. We recover here. We recover here in 15 minutes, okay? Then we have the short session on regulation and psychology, and then we have the round table, okay? So 15 minutes for your break.